Hello guys, welcome. So today we're going to speak about it's a DNA and RNA. So today we're going to elaborate, get more information about the RNA and the DNA. Okay, so we have the monomer known as a nucleotide. The nucleotide it's made up of the three components, which is the phosphate, nitrogenous base, and the pentose sugar. It's also known as a five carbon sugar. So the five carbon sugar depends on the structure. It's either the DNA or the RNA. For the DNA, it is a deoxyribose. For RNA, it is the ribose. Now we go to the nitrogenous bases of the DNA. You've got adenine, thymine, guanine, and cytosine. Then for RNA, we've got adenine, uracil, guanine, and cytosine. So the difference between the two is that the DNA got the thymine, the RNA got the uracil. Here's the deoxyribonucleic acid structure, which is double-stranded. We've got the many cycles here. All these cycles are known as the phosphate group. Okay, then we've got the ribose sugar. You see, deoxyribose sugar. Okay, you're going to get it on that way. Then we've got the nitrogenous base. The nitrogenous base it can be guanine, cytosine, adenine, and thymine. Then we've got the bonds in between the nitrogenous bases. The bonds are different. So between the cytosine and the guanine, we've got three bonds. Then between the adenine and the thymine, we've got two bonds. That means the bonds between the guanine and cytosine are stronger compared to the bonds between adenine and thymine. Then we've got the phosphodiester bond. The phosphodiester bond, it is a bond that is linked to or more nucleotides. Okay, so the nucleotides that at the back, which known as the sugar phosphate backbone, and that way we find our phosphodiester bond. Okay, then let's go to the ribonucleic acid structure. It is single-stranded structure. There is a phosphate group, those cycles, same as the DNA. Then there is a phosphodiester bond, the nucleotide and the sugar phosphate backbone. Then we've got the ribose sugar because it's a ribose sugar. Okay, then we've got the nitrogenous bases where you find the cytosine, uracil, guanine, and the adenine. Okay, then we've got differences between the DNA and RNA. There are more differences, but I'll give you guys these three differences. Okay, then we've got a double-stranded. DNA is double-stranded. Okay, RNA it is single-stranded. Then the DNA got the deoxyribose because it's a oxyribonucleic deoxyribose sugar. Then RNA got the ribose sugar because it's a ribose. Okay. Then we've got a DNA, there is a thymine, while RNA there is a uracil. Let's go, guys. Here we've got the DNA replication. The DNA replication, it is a process, okay? The process that is lead into the production of the two identical daughter cells, okay? So there are some processes or some steps that must be followed in order the DNA replication can be successful. Okay, start with unwind, unzip, then form the template, then there's a complementary strand form. Okay, let's go into details. The DNA take at the interface before the mitosis begin the process of this DNA replication. So the double helix unwinds. The weak hydrogen bonds between the nitrogenous bases break and two-strand unzip. Okay, there's this enzymes known as a helicase. The helicase is used to unzip the hydrogen bonds between the nitrogenous bases. Then after the unzipping, we know that they are separated now. We've got two strands. Then each strand serves as the templates. So the free nucleotides get attached into the original DNA template, which is the single strand, then this may result into two identical DNA molecules. You can say it's a daughter cell. So there's a significance of DNA replication. It results into the formation of identical daughter cells. Okay. Then we've got a DNA profiling. The DNA is profiling, like DNA is very important, guys. It used to identify the paternity, their bodies, relatives, criminals. Okay, that is very, very important. We've got the protein synthesis. The protein synthesis, it is the process of how the DNA produce the proteins. Okay, then okay, let's start. There is a transcription. There is a translation. Transcript C comes before L. So let's start with the C. Okay, so it's first the DNA and one is split into two strands serve as a templates. Okay, then the free nucleotides get attached into the original strand of the DNA, which is the templates. That's known as the transcription. Then the mRNA leaves the nucleus. It will leave the nucleus through the nuclear pores. You know that the DNA, it's located in the nucleus only. It can't move around the cytoplasm. But because the mRNA can be located into two locations. It can be in a nucleus, it can be in a cytoplasm. Then the mRNA is used to transfer the information from the DNA into the ribosomes. Okay, so as to continue to add, the nuclear pores. Okay, each tRNA brings a specific amino acid to the cytoplasm. Then those amino acids get bonded together by the polypeptide bond. That's how the translation occur. Okay, as you see here in that picture, these amino acid that is scattered, then there's a polypeptide or the peptide bond that's linked them together to form a protein. Thank you for watching, guys. I will appreciate your subscriptions, and there'll be more information I will upload.